Our next speaker is Dr. Manuela Dal Forno. Manuela is a lichenologist at the Botanical Research Institute of Texas, and her work focuses on multiple aspects of the lichen symbiosis. Following the completion of her doctoral studies, Manuela served as a National Science Foundation postdoctoral research fellow, conducting collaborative research at the Smithsonian Institution's National Museum of Natural History and the University of Graz in Austria. Subsequently, she was awarded the Peter Buck postdoctoral fellowship here at the NMNH, where she furthered her investigations into collections-based research with what she says, and I believe her, is the coolest group of organisms, the lichens. So please join me in welcoming Manuela.
Hello, everyone. Thank you, Dr. Johnson, for the introduction and for the invitation to be here. I'm really excited to be back and see so many familiar faces. I would also like to thank everyone involved in the organization of this very special event. And my name is Manuela, and I'm really, again, excited to share with you some of the results of my research that I've done while I was a postdoc here at the museum. Starting with, what are lichens? Lichens are a multi-species symbiosis, and symbiosis literally means living together. Only when you look really up close in a lichen, you are able to see that it's composed of cells of a fungus, a photosynthesizing organism, that it could be a green algae and or a cyanobacteria, along with a community of microorganisms. So I really think that we can study lichens in the context of fungal systems, photosynthesizing systems, as well as microbial systems. And to add to this really complex world of lichens, they do not come all from the same common ancestor. Lichenization is a fungal lifestyle that is based on nutritional mode, along with decomposers, parasites, and so forth. And this habit, lichenization, has evolved multiple times throughout the fungal tree of life, and independently in Ascomycota and Basidiomycota, which are the two main groups in fungi. There are currently about 20,000 species of known lichens, and that means fungi that have evolved to this lichenized habit. 99% of these lichens, of these 20,000 species, are Ascomycota, and only 1% are basidiolichens. And this is what we're going to talk about today. And just by looking at these at first, a lot of people don't even realize that some of these are lichens. And I'm going to focus especially on the subtribe Dictyonemmacini that are not only basidiolichens, but they are also the only group that is a basidiolichen and a cyanolichen. So they also have cyanobacteria as their photobiont. And they are found worldwide, but have their highest diversity in the tropics. And historically, it's all been known to be one single genus, Dictyonema, and 5 species. But we were out there in the field and looking at all of these different morphologies. So our studies really started with the basic question, how diverse is this group? And our studies started about 15 years ago, what used to be one genus and 5 species, to what we know now as 160 species organized in 5 different genera. And we were able to resurrect a lot of these names, and with our continuous studies, we have been able to identify detect already 450 species and estimate over 800 species that are belonging to this subtribe. And it's important to mention that this is not only done with genetic data, but also combining ecological data and morphology to come up with these hypotheses. And there is still a huge discrepancy to how many species we have detected to how many are estimated. So we started to wonder, how can we fill this gap with herbarium samples? And this is how I get to this wonderful place, the U.S. National Herbarium, here at this museum. And lucky for me, it holds the 2nd largest collection of Dictyonemmacini in the world, spanning for throughout the 1900s, and I've decided to test different assessments of biodiversity. People always ask me, how do I study the genetic of lichens? Because there's just so many things going on there. And it's, well, there are quite a lot of ways, actually. And just like other groups, a lichen is a natural environmental sample, so you can select which particular group you're interested, or just sequence everything. And I always say that choosing the right methodology, I guess for any of the organisms that we study, is very important to choose the right technique to address the questions that you are interested. And due to time restraints today, I'm just going to focus on these 3 methodologies today that, of course, I'm going to go into a little bit more detail. To get started, so people can explain DNA sequencing in a lot of different ways, and for the purpose of this talk, I'm going to simplify the techniques, the sequencing techniques that I utilize here in high-throughput sequencing, is also known as next-generation sequencing, is how we can generate a lot of data, so sequence from the DNA that we know as reads, so for a lot of samples at the same time. And there are 2 main techniques, the amplicon sequencing here represented by metabarcoding, that we're able to select a specific region of the genome that we're interested in studying, and that can be one or few loci. And there's shotgun sequencing, that you just sequence everything that is in the sample, so theoretically you could get all the genes belonging to all organisms in that sample. And starting with the metabarcoding, for fungal diversity, we utilize ITS, and barcoding is an idea that all living organisms have a unique sequence of DNA that we're able to identify all living organisms on Earth, and it's similar to when you go to a grocery store or a retail store, that you purchase a product, and when you go to the cashier, it will scan the barcoding. So it's a similar idea. For lichens, I start with a little piece of sample. I can get very little and already generate a lot of sequences. So you have the sample, you extract the DNA, and you sequence the barcoding. And I have to say that I know that there's no one-size-fits-all. This works better for some groups than others, but gladly it works really well for species delimitation in this group. And gladly, too, most of the reads, the sequences that I get with this method, belongs to the main fungal partner. So it's very easy to identify which reads are belonging to these. However, these methods are so sensitive that sometimes you also pick up sequences for spores that are just landing on your lichen sample. For these results, here we can see a graph that in the y-axis is the percentage of success rate that we had. And in the x-axis, or the horizontal, it has the groupings. And for a total, so considering the entire subtribe, those 418 samples, we were able to get 75% of those samples. We could get an ITS sequence. And here are the raw numbers in pink and in green. And then I also wanted to see if the foliose forms right here are performing similarly to the filamentous forms. And next, now I'm going to focus on the foliose forms, because historically they've only been known as one name, Dictyonema glabratum, or Cora glabrata, or Cora pavonia. And a lot of my colleagues up here have already explained the concept of a phylogenetic tree. And then this is just made in circular. And it infers the relationship of all of these samples, and because we've been working with this group for such a long time, this tree actually has 1,325 terminals, that all of these tiny little lines that you see around, and all of those in blue belong to specimens from the US herbarium. So keep in mind the pattern that you're seeing now. And this is slide that I'm going to present some of these key findings. And the map that you see is the natural distribution of this group of lichens, Cora, the foliose forms. And you can see that all of those in yellow are samples that we didn't have samples before. And we were able to add 11 countries to our sampling that we didn't have before. And just looking at a few examples, not even going at the state and region level, but at the country level, Panama, for example, we had a single specimen that we identified as one species, but while adding dozens of specimens from the US National Herbarium, now we know that at least historically, Panama has had at least 6 species of Cora. So it's a huge impact in the numbers of biodiversity. And to give an example of a species, Cora itabaiana, it's really fascinating because it actually lives in a dry environment, so we were really surprised to find this sample there. And then again, we only had a single sample from northeast Brazil, and adding the herbarium samples from here, we were actually also able to find this species in Suriname, Guyana, and Trinidad and Tobago. And lastly, what I found one of the most incredible outcomes is that all of these 275 Cora, so just the folio specimens, they were all filed under one species, Dictyonema glabratum, in the herbarium. And now, when I'm gonna put those back in the shelves, they're actually gonna be representing 75 species in 2 genera. And out of these, 25 are new lineages, so discovered entirely from the herbarium here. Switching gears, I'm jumping to metagenomics, and now I will explain this utilizing the analogy of a puzzle. Think of an organism as being an entire puzzle put together. And when you perform shotgun sequencing, you are fragmenting the DNA into millions of pieces so it can be sequenced by these available technologies, which is your puzzle pieces. And now imagine that a multi-species organism like a lichen, we call it metagenomics because it's like you have a lot of different puzzles in the same box. So you not only need to figure out where the puzzle pieces go, but also to which one of these puzzles it belongs to. But eventually, you know, there are different ways that you can do that in different orders and bioinformatic pipelines that you can assemble it, but eventually you might get a couple of pieces missing. I wanted to test if herbarium samples could provide good DNA for this type of analysis, because in general, they are sequenced with fresh specimens. And during my Peter Buck Fellowship here, I sequenced 13 genomes, and a couple years after, or the last time that I visited, I thought, how about we also add some historical samples? And this is a quick overview of just a few species that we were able to finish our analysis. And these are the 2 samples that we sequenced for Cora, one fresh and one older. And same thing with Dictyonema down here at the bottom. And for those interested, this is the genome pipeline that we use, and the finalized product is the genome assemblies that we're using to assess this Bosco that I'm gonna walk you through. So a quick comparison utilizing this software known as Bosco, which utilizes data sets that are nearly universally distributed single copy orthologous genes within the species at a certain phylogenetic level. So we can, for example, test which genes are present in these samples that we sequence at the fungal level, and also because these are basidiolichens at the Basidiomycota level. And now I'm going to call attention to a couple of these numbers. Here you see N758. So that's the number of total genes that this software is utilizing to compare your sequences to. So same thing here in Basidiomycota. It was actually searching for nearly 1,800 genes in these genomes that we fed into this software. And we can immediately see that the oldest sample here, the second one, Coratimocua, has the most red. And red, if you look in the legend, says for missing genes. So we were not able to get a lot of genes out of the sample, but we all, or the people that do work with genomes, know how much these analysis highly depend on the quality of the data and also the quantity of the data. So it's possible that I just need to repeat this, but I'm still really excited because look at the bottom sample, the Dictyonema US 166. It's a new species from Australia, that it actually outperformed the fresher sample. So there is a lot of potential, and I'm not sad at all that Coratimocua didn't perform as well, because it still provides me with 51 single copy orthologous genes that I now can infer relationships, while before I had much less data. And what is fascinating to me is that when I first sequenced these genomes, I had no idea of the new questions that I would be able to answer when I started this project. And I recently started working with this colleague, Paul D'Agostino, who's interested in biosynthetic gene clusters. So he is the one that is doing a lot of this analysis. And this is particularly important in the context of natural product discovery and their potential use for biotechnology and medicine. So he's mining for these genomes of lichens that I generated years ago for something else that he's interested. In addition, this data has also been utilized by 2 PhD students in their studies with cyanolichens. And lastly, I wanted to show that another way to explore microbiomes is with fluorescent in situ hybridization that I had the opportunity to learn as part of my postdoc. And it starts with your fresh sample, and then you add probes that have fluorescent dyes. They will hybridize to the DNA of your sample, and then they will light up if the phylogenetic group that you are selecting is present. And the parts that you see that are brighter are a probe for eubacteria. So the larger cells that you see is the photobiont, while the tiny other dots are other bacteria that are there. And this is the beauty of fish, that you not only see what is present, but where is present. And for the first time, we performed fish with basidiolichens and with historical samples. And these 2 that you see on your left are samples from 1981, and now I'm comparing that same species to a sample that I collected recently to see the patterns of bacteria in these lichen thalli. So in conclusion, I like to think that all of these techniques are really wonderful to add new ways or enhance the value of existing collections, and especially the metabarcoding that we had really positive outcomes. It's really important because there are some very challenging groups that you identify lichens solely on herbarium samples, because a lot of them lose their characters. There was even a question about that, about the color. Lichens, on the other hand, do lose a lot of their characters, so adding genetic data is really important to have accurate identifications. And as a last example, my colleagues and I started noticing that one of these coras that historically has only had one name, this species from Florida, had a very unique phenotype. We got them all on loan, the 32 existing cora from Florida, and we were able to sequence both samples from the early 1900s from the US National Herbarium. So you see on top over there, Coratimocua. So we proved it was a new lineage from Florida, and we published as a new species discovered from herbarium samples. And we even got some PR. And this is really positive. Well, lichens in the news is always positive, but also because we noticed that our last collection of in all over the world, all of these samples from Florida, the latest one is from 1985. So even though we discover a new species, is also potentially already extinct, so we were able to list as critically endangered under the IUCN Red List, and we also have an ongoing iNaturalist project to potentially track these species in the wild. So to finalize this talk, I just wanted to say that my current hypothesis is that 50% of the unknown, so the huge gap of knowledge is still in this group, can be found in herbaria. And these are some lists that I've collected in the 2 major portals that have lichen specimens deposited on them. And I like to think that herbarium is still going to help me find the other 50%, because this way I can see what areas are the highest diversity of these lichens, where there is poor sampling, and also see if there are some of these areas that are potentially threatened by habitat loss and climate change. So these collections not only offer a window to the past, but they also guide us where we should focus our future efforts to advance biodiversity knowledge. With that, I would like to thank all of my colleagues, as well as multiple funding sources throughout the years, and I'm happy to take any questions.
Questions for Manuela?
Questions? Mm-hmm. So Rebecca's question was if the composition of the lichen changes over time, especially for historical samples. And no, the lichen is able to maintain its main shape in the herbarium, but it does change because it loses a lot of the water content, so the lichens get dried up, and that may result in the changing of colors. So that is one of the first things that I see when I was selecting material for these, that some of them are able to maintain their full color. It seemed like it was collected yesterday, while some become entirely brown, so you don't see the beautiful color of the cyanobacteria anymore. However, the chemical compounds present, they remain there, so we can still utilize that data. So that doesn't change. Thank you. June?
Hi, Manu. Fantastic talk. Thank you. And you predicted about 50% of the diversity of these groups are in the herbarium. Do you have any idea about how much unknown diversity needs to be collected? I mean, you have some data, I think, in the Philippines. Yes. So from that type of data set, what would you predict about the unexplored diversity which have never been collected?
I think that's a great question. I've thought a lot about that, and I think, and I might be totally wrong, but I think that maybe we know about 5 to 10% of all lichens currently, so the 20,000. The current estimates of fungi is millions, so taking that into consideration, I think that there might still be, 3 to 500,000 of lichens. Yeah, 3 to 500,000 lichens still to be discovered, and most of them, or at least a lot of them, are still out there, so we really need to run to discover those before they're gone.
250% undiscovered.
Yeah, well, that's, I mean, I'm sure, because it's, I know saying a hard number is difficult, but I'm 100% sure that is more than like 10 times at least the unknown diversity, especially for lichens, because anywhere that we go, especially in the tropics, there's always lots of new species, dozens of new species, and you see papers that come out with 100 new species, 1,000 new species of lichens. So I definitely believe that there's still a lot more out there. So come work with lichens.
Thank you.
Thank you.
How
Yes.
long does it take for you guys to figure out the genetic coding and the materials in the different types and diversity of lichen?
Sorry, can you elaborate a little bit more? How long does it take for like the whole pipeline?
Yes.
Okay, well, I would say that these days that there are some library preps that you could literally start from sample to genome sequencing in one day. So when I was here, let's see if I see some lab people here. So our DNA extractions, it can take, the one that I use for the genomes takes, for a few samples or for 24 samples, it takes about 3 hours. And then the library prep too takes about 2 or 3 hours, and then you do some cleaning. So you can do all of that in one day and have your sample prepared for sequencing the next day. And we have a great lab manager here. I can see by the big light. And she sequenced everything for me and for a lot of other projects very quickly. So you can have your data from start to finish, having your data in less than a week. So it's really amazing how far we've come and how you can really get a lot of data in very little time. It's a great question. Thank you.
Thank you.
Julia.
If you're identifying new species of lichen, are you also identifying new like bacteria and fungi strains within those? Like how do those?
Great question. And when we talk about new lichen species, or for example, June's question, these numbers are just based on the microbiome, so the fungal partner, the fungi that have evolved to become lichenized, and now depend on that exclusively. If they don't find a partner, over 99% of the lichen will not survive. There are very few exceptions. So when we talk about new species, we do discover mostly from the fungal component. However, over the past 10 years, there has been a lot of research also done from their photobiomes. So there's also a lot of new species that are describing new cyanobacteria, new green algae, and also people that discovered also in bacterial lineages that are entirely associated with the lichen symbiosis. So you can really discover biodiversity at all different levels. Thank you.
Manuela absolutely convinced us how cool lichens are.